We will have more time later on when discussing the general topics. Then thanks again. And it's my pleasure to invite Grossman from Teva Pharmaceutical Industries, and there is something in between.
Thank you, Dan. It's a tremendous privilege to speak today on this topic after Professor Seller's keynote speech this morning. He shared with us the inception of Glatiramoids and Copaxone specifically, and I consider my talk today a downstream cascade of that. I represent Teva Pharmaceuticals, and today I would like to share with you data on the specific subgroup within non-biological complex drugs that constitutes glatiramoids. And to introduce Copaxone, as Professor Sella mentioned earlier today, it's a complex synthetic mixture of polymers consisting of 4 amino acids at a predefined ratio. As I will describe in detail today, the active moieties are unidentifiable, and therefore the entire mixture is needed for the therapeutic effect. It is a subcutaneous injection, and the drug is immediately hydrolyzed upon injection. Therefore, there is no measurable pharmacokinetic profile. Similarly, there are no validated biomarkers of the pharmacodynamic effect of the drug, and again, I'll go into that into more detail in several slides. It is an antigen, and therefore it has immunogenic potential. However, strict antigen homology has been ensured through more than 2 decades of patient experience with well-characterized safety profile. And so I'd like to share with you a high level of the synthesis process for Copaxone to describe its compositional complexity. So the first stage includes polymerization under strictly controlled conditions from the 4 activated amino acids, monomers, and then the product of that stage is protected gladimer, which is a high molecular weight polymer of thousands of amino acids long, each with a different sequence, and that is the gemisch of that end product for the polymerization stage. Now, the second stage takes this high molecular weight polymer and cleaves it into shorter fragments of up to about 300 amino acids per fragment at different sequences. And so, combinatorically speaking, this yields up to 10 to the power 29 different variants, different sequences in the mixture. I'm not going to go into great details of all these factors that are mentioned here and others that are not, but each one of these stages is affected by a variety of different factors, each subtly, potentially subtly changing the composition of the end product, which could lead to differences in the therapeutic effects. And so it is for these reasons that Teva has not modified any of the critical parameters that affect Copaxone's composition since the pivotal clinical trials in the early 90s. And so Copaxone is clearly more complex than small molecules, but it is in fact more complex than many biologics, and certainly from all approved biosimilars to date. In fact, if you will, Copaxone mixture includes thousands of different proteins within the mixture, because each one of these polymers is a backbone of its own. Because each at a different sequence, different size. And because there are so many variants, as I mentioned earlier, each exists at sub-picogram quantities in the mixture, which means that currently available state-of-the-art methodologies cannot be used to fully identify all of these variants. We cannot use bottom-up or top-down sequencing. And so, for instance, infliximab is far more simple, or it's less complex than Copaxone. The backbone is fully characterized, and the variation is dependent on the glycosylation pattern at specific residues. For the rest of my talk, I will share with you experimental data that we have accumulated over the years internally and with collaborators, which characterize first and foremost, of course, Copaxone itself through sometimes dozens of different batches at a time, and then all the marketed drugs that are intended to be follow-on glutaramyr acetides. So I will use the acronym FOGA in the next slides, have been characterized by us as well comparatively. So these are products from India, Mexico, and Argentina. And the theme that will be the major conclusion, or one of the major conclusions from my talk, is that when we apply simple, traditional, low-resolution methodologies, be they physicochemical in nature or biological, some similarities are seen between different compounds. However, when we use multidimensional, high-resolution methodologies, marked differences are seen, and some of them allude to potential clinical relevance. So just two quick examples for methodologies that show similarities. Here is size exclusion chromatography, and in all my slides you'll see Copaxone in blue, and all the various Glatiramoids I mentioned earlier, differently manufactured, are in red. And you can see profiles seem quite similar. That is also true for circular dichroism. Now I will show you one example. We have multiple other methods that show a very similar result to this one, but I'm going to show only this one for the shortage of time. We have published and submitted this information previously. So here is ion mobility mass spectrometry. It's a method routinely used for separating or characterizing peptides, proteins, and the key attribute is that a compound or a mixture can be simultaneously characterized for its shape, charge, size, and amino acid sequence at the same time. So here on the right, you have a two-dimensional picture of one such compound characterized. Now, if you want to compare two different batches or two different compounds, you virtually overlay the heat maps of these two compounds, and you seek differences. So on the bottom left is a comparison heat map of two compounds that are absolutely 100% identical. And you see no differences, no color, it's all black. On the bottom right, you see significant differences between the two compounds depicted by bright colors. The brighter the green or the red, more differences, and these colors represent directionality. If you want to compare many different such experiments of comparison heat maps, you often summarize the entire differences in the picture into total intensity values, and then I will show you later, you can compare those. I want to emphasize the point that IMMS lets you evaluate differences at the pixel level. So it's a very high resolution methodology. So here, of course, first we characterize Copaxone. These are just a few examples of interbatch differences. And you can see that there is minimal inherent microheterogeneity that we expect from complex medicines, but it is minimal. If we compare the exact same Copaxone batch to any one of the FOGAs, and here I'm only showing 3 of those, marked differences are seen. And then if we look at the TIVs, of all these comparisons, what you see in blue on the left are 20 different batches of Copaxone, all showing a minimal total intensity value range, which again represent microheterogeneity within the compound. On the right, we see 8 different samples that represent, or comparisons, that represent 4 different phogas, which means that for some of them we have various batches in the picture. And first and foremost, we see the entire range is orders of magnitude higher, and there is also inter-batch variability, which represent significant compositional differences. Next, I'd like to talk about the functional characterization of these compounds. And briefly, I'd like to give you an overview of the mode of action of Copaxone, which in fact we keep on elucidating every year. New pathways are identified, and it's really yet unclear what is critical, what is sufficient, and exactly how it works. We do know that once hydrolyzed, the fragments are being taken up by antigen-presenting cells, which present them the fragments to T cells. They become Copaxone reactive T cells and induce a shift from a pro-inflammatory phenotype, the Th1, Th17, to an anti-inflammatory phenotype of Th2, as well as T regulatory cells, which are marked by FOXP3. And also non-neutralizing antibodies are produced, which are considered to be associated with positive response. Then these reactive T cells cross the blood-brain barrier and again induce anti-inflammatory milieu and also induce the secretion of neuroprotective factors. And so with all the fantastic work that has been done over many, many years to elucidate Copaxone's mode of action, it's still, the traditional methodologies are still insufficient. And therefore, we have applied genome-wide expression profiling, which allow us in an unbiased way to look broadly and comprehensively across the genome and test many different cell types and model systems. And this is to show that the results when we characterize Copaxone show hundreds of genes affected and relevant pathways affected, validated by prior experimental data. Then we applied various different experimental data, designs to better understand Copaxone and then compare to Glatiramoids. Here, this is one experiment in which we aim to model switching or interchangeability. We see marked differences in FOXP3, which I mentioned earlier is important for the mode of action of Copaxone and specifically for MS disease severity. We then also we ran the same experiment with a reciprocal design to ensure that the differences we capture are drug-specific. And indeed, hundreds of different genes were affected. They were related to known functional relevant disease-related activities, such as IL-18. Then we also modeled the antigen-presenting cell side of the immune response. Hundreds of genes, again, were affected, including pro-inflammatory pathways that were upregulated significantly by the FOGA compared to Copaxone, specifically LPS, which is a prototypic pro-inflammatory pathway, I'm sure many of you know. And that is concerning because adverse events leading to discontinuations with the same drug were reported from a clinical trial compared to Copaxone. And the same pattern has been seen by us previously with Probioglat in Mexico. Again, in the same experimental model, that drug induced pro-inflammation, including LPS, and then patients reported increase in adverse events and relapses upon introduction of that drug to the market. To further validate what we found in other biological systems, in other technologies, we ran protein secretion panels. We found confirmation to our microarray findings for both drugs. And then we also looked at primary monocytes from human donors to make sure that the THP1, the cell line monocyte results, were indeed related to the drug and not to the transformation itself, and that was confirmed. And so to summarize with my conclusions, Copaxone is a complex drug in terms of its compositional structure as well as mode of action. It cannot be fully characterized at present. The active moieties are unidentifiable within the mixture. Some similarities are seen when we use low-resolution methodologies. However, marked differences are seen both in terms of physicochemical as well as genomic and functional characteristics. And lastly, Genomewide expression profiling reveals significantly different effects on the activation and regulation of immunologically relevant pathways. We see that in different experimental models and cell types, including human and mice, and that was further confirmed in the protein level as well as in primary monocytes. The upregulation of pro-inflammatory gene expression might explain some of what we see in the clinical settings. I would like to thank you for your attention.
Time for one short question, short answer. Yes, please. Well, I think you had your turn, so who else is there? No, I think you have the freeway. Short question, short answer.
Short question. The changes in the cytokine pattern that you show from the Th1, Th17 to Th2 and Tregs are specific for any antigen, are myelin basic protein, or are specific for any of the peptides that they are included in Copaxone?
So I'm not sure I fully follow your question.
So you measure T regulatory cells and Th2 cells. What are these T cells recognizing, or is this just the global pattern?
So we don't measure that. If you relate to the body of evidence out there, we see these patterns, and it's not yet clear specifically what is the autoantigen when these T-cell Copaxone reactive T cells enter the brain, whether introduction to MOG or MBP or PLP, which one of them induces the exact specific pattern of pro-inflammatory and inflammatory cytokine release in the brain. So the exact autoantigen reintroduction is not fully understood. Did I answer your question?
More or less. What I imagine is that you take and
I can talk. Okay, sorry.